Hello, today I'm going to be discussing lysogenic conversion. To begin, the term lysogenic signifies that prophages are capable of generating active phages that lyse their host cells. Thus, lysogenic conversion is a change or conversion in the properties of a bacterial cell as a result of its infection with the temperate bacteriophage. It induces a change in the phenotype of the bacteria infected that is not part of the usual phage cycle. This lysogenic cycle allows replication of the phage genome without destroying the host. Lysogenic conversion occurs when a lysogenic bacteriophage containing an exotoxin invades a bacteria, thereby giving the bacteria the ability to replicate and express the same exotoxin copied from the lysogenic bacteriophage. This happens as a result of the integration of the bacteriophage's DNA into the bacteria's host chromosome. Then, the integrated DNA prophage from the bacteriophage enables the bacteria to possess a new genetic trait which specifically allows them to code for the protein exotoxin or other virulence factors, making the bacteria more dangerous. Next, I'm going to be highlighting three differences between lytic and lysogenic replication of bacteriophages. The first difference is in the viral genome replication. In lytic replication, the viral genome replicates independently of the host or independent of the bacterial genome replication. However, in lysogenic replication, the viral genome replication is completely dependent on the replication of the host bacterial genome. After injection of viral genome in lytic replication, the genome remains free in the cytoplasm, so there is no prophase formation. However, after injection of viral genome in lysogenic replication, the genome gets attached to the bacterial genome to generate prophage. Lastly, lytic and lysogenic replication of bacteriophages differ in that lytic replication causes lysis of the bacterium after the immediate replication of the bacteriophage. Lytic phages are called virulent phages, completely taking over the cellular mechanisms of the host cell, and this includes T phages. Whereas in lysogenic replication, there is no immediate lysis of the bacteria after replication, so it is called non-virulent or temperate phages as it only slightly disturbs the cellular mechanism of the host cell, and this includes lambda-phages. Lytic bacteriophages cannot follow the lysogenic cycle, while lysogenic bacteriophages can follow the lysogenic cycle until it is induced to enter the lytic cycle, and this is known as lysogenic conversion, as I mentioned earlier, which carries genes that enhances virulence of bacterial host cells. Now I'm going to explain the role lysogenic conversion plays in the development of pathogenicity in bacteria. When a temperate phage, such as a lambda phage, generates a change in the phenotype of the bacteria infected that is not part of a usual phage cycle, the changes can often involve the external membrane of the cell by making it inaccessible to other phages or even by increasing the pathogenic capability of the bacteria for a host. Lysogenic conversion plays a significant role in the development of an increased pathogenicity of a bacteria because the added genes expressing exotoxins or other virulence factors from the bacteriophage enhances the ability of the bacteria to cause pathogenic effects or virulence. This is caused by the integration of the viral genome into the bacterial DNA, wherein the bacterial DNA gains the genes that expresses exotoxins or other virulence factors, such that when the bacterial DNA replicates, the ability of the virus to create exotoxin is passed onto the daughter cells of the bacteria, creating new bacteria that now has the exotoxin. This process enhances the pathogenicity in a bacteria, thereby making it more dangerous. Lastly, I will provide examples of where lysogenic conversion has contributed to pathogenicity. Vibrio cholerae, a gram-negative bacteria causing cholera, which causes severe dehydration and diarrhea to humans. According to history, V. cholerae has not always caused these signs and symptoms to humans, but due to the lysogenic conversion occurrence where the CTX phage had integrated its genes to the V. cholera, resulting to cholera toxin, which is an exotoxin, thereby causing cholera to humans. Another example is Corynebacterium diphtheriae, a gram-positive bacteria causing diphtheria to humans, where the same occurrence of lysogenic conversion due to a bacteriophage called corinophage beta, where bacteriophage's genes have been integrated into the C. diphtheria DNA, resulting in the ability of the bacteria to create the diphtheria toxin, which is also an exotoxin, causing diphtheria to humans. In some cases, lysogenic conversion enables bacteria to form biofilms, like the bacteria Bacillus anthracus. And due to the formation of biofilm bacteria, it gets better access to the nutrients and to survive bacteria in harsh environmental conditions. Lysogenic conversion also enhances sporulation that converts vegetative bacterial cells into dormant states, that is resistant to radiation, temperature, and resistant to antibiotics and desiccation. Two more examples that I thought I'd mention. The bacteria Clostridium botulinum causes botulism, which is an example of lysogenic conversion. Additionally, Streptococcus pyogenes causes scarlet fever. And that is it for today, you guys. Thank you so much.